Hey there, and welcome to section 2 of this course, Reproductive Anatomy and Physiology. In the last section, we covered the fundamentals of artificial insemination, including its definition, historical evolution, practical benefits, and key challenges. Now that we understand the why, it's time to explore the how, starting with the biological systems that make reproduction possible in livestock. In this video, we're going to learn about the male and female reproductive systems of cattle, goats, and sheep. Understanding these structures is essential. Why? The success of artificial insemination depends not just on the quality of semen or the timing of insemination, but also on how well you understand the animal's reproductive anatomy and how to work with it, not against it. Let's begin with the male reproductive system, which includes testes. These are the primary reproductive organs. They produce sperm and the hormone testosterone. In bulls, bucks, and rams, the testes are located outside the body in the scrotum, which helps regulate temperature for optimal sperm production. Epididymis. This is where sperm mature and are stored before ejaculation. Vas deferens. These tubes transport mature sperm from the epididymis to the urethra. Accessory sex glands. These include the seminal vesicles, prostate gland, and bulbourethral glands. They produce fluids that nourish and protect the sperm. Penis, the organ that delivers semen during natural mating, and where the semen collection process begins during artificial insemination. Now let's turn our attention to the female reproductive system, which includes ovaries. These produce eggs and reproductive hormones like estrogen and progesterone. Oviducts, also known as fallopian tubes. This is where fertilization happens, where sperm meets egg. Uterus, a muscular organ where the fertilized egg implants and develops into a fetus. Cervix, a tightly sealed passage that opens during estrus and parturition. This is a key structure when performing artificial insemination. The insemination straw must pass through the cervix to deposit semen in the uterus. Vagina and vulva, these serve as the reproductive tract's entry and exit points, and they also play roles in mating and birth. Now, while the basic structures are similar across cattle, goats, and sheep, there are slight anatomical variations you should be aware of, such as the size, length, and positioning of the reproductive tract, especially the cervix and uterus. For example, the cervix in sheep is more complex and folded than in goats or cattle, which can make insemination more technically challenging. Understanding these species-specific differences helps you adjust your technique and improve success rates. Reflection questions. Take a moment to reflect. Have you previously examined or worked with the reproductive system of livestock? What challenges did you notice? How might a deeper knowledge of anatomy improve your ability to perform artificial insemination? Why is it important to understand differences between species when applying the same reproductive technique? In the next video, we'll move from structure to function, exploring the estrus cycle and how to detect signs of estrus, the fertile window that makes or breaks the success of artificial insemination. Let's keep going, and I'll see you in the next one.